Thank you, Bjorn, for inviting me, and it's an honor to be here, especially on this day, the 194th birthday of Theodore Billroth. So in my talk, I'll be talking about the secrets of shapes in nature, and in particular biology, and how that is relevant to medical practice. So to start, I like to talk about why we study shapes in nature to begin with. In biochemistry, the mantra is structure is function, right? And that means that when you have recurring patterns of distinct shapes, right, you will find recurring patterns of distinct functions that correlate with those shapes. But that mantra is true also throughout the other larger scales of biology. Of course, in clinical practice, the phrase is more like structure is clinical outcome, which is why fields of pathology and oncology and radiology exist, right? We interpret the shape, the borders of the tumor, and that helps us to, grade it, whether at the macroscopic level or at the, immunohistochemical level. Take, for example, these two coffee mugs. They're the same. I just inverted one of them. So both of these mugs can hold the same amount of hot water, but if you measure their volume, right, which is a popular approach to measuring, liquids, right, or three-dimensional objects, volume will tell you that these two coffee mugs are the same, meaning volume cannot tell you which hand is about to be burned with that hot water. And the reason is because volume and area and perimeter, right, our go-to methods for measuring spatial information, these methods are agnostic to spatial relationships within a biological system. But the thing is, spatial context, right, exists at all scales within nature. And we know this intuitively, right? We learned this in school in our training, but it's easy to forget this because our go-to methods, such as volume, area, surface area, are all agnostic to spatial context, right? So in panel A, as a reminder that even amino acids at the molecular level, they are left-handed or right-handed. On Earth, they need to be left-handed in order to be incorporated into life as we know it. And yet there is no objective mathematical way to define a left versus right-handed shape, right? And when I say define that, I don't mean shining light onto it like spectroscopy, right? That's an indirect measure of chirality, left versus right-handedness. But in terms of shape, we don't have a method, but we do now, right, through the technology that I'll be describing. And in panel B, you can see that even with gland structures, they follow rules when they develop. And when they don't follow rules, they push into each other, they invade each other, and we call that cancer, right? So there's spatial context important in development and disease. And even at the level of ecology in panels C and D, right, we see evidence of spatial context, right? In panel C, there's a phenomenon known as crown shyness, that is trees, certain types of trees, make sure not to crowd each other out. So in order to fully understand the growth of one tree, you have to understand the position of its neighbors. And then panel D, moss and lichen, they like to grow on one side of trees. And it's no longer just the north side as it was previously believed, but now it's this, we know that they grow on the side that has the least direct sunlight, right? They're trying to avoid, the most direct sunlight. So even at the level of ecology, there's spatial context. So I developed an algorithm which we turned into a software called Shape Genie, and I will describe how it measures shapes because we applied this technology to all of the clinical research examples that I'll be talking about in this presentation. So the key innovation, right, of Shape Genie is a grid system that turns two-dimensional objects into a sound wave, right? We call it a sinusoid pattern, right? Which looks like a sound wave. And so there are many grid systems, but the two common ones that we use are the parallel grid system and the radial grid system. So on the top row, the shape of interest is a crescent, right? Or it could be a sickle cell. And we apply, in this case, a parallel grid system of parallel horizontal lines. And that allows us to, as you see in panel B, find every intersection between the grid line and the object in blue. And once we do that, we can measure the distance that every dot, the blue dot, makes to that dotted line, which is the reference point from which we measure distance. So in this way, we can turn a two-dimensional shape like this crescent into a sound wave, essentially a sinusoid wave. And in the bottom row, we use a different grid system. We call this the radial grid system. That is, we define the center of the object as the origin of this radial system. And we apply a grid of 360 degrees every 5 degrees, worth of radial lines. And we can again measure the distance of every single intersection between the radial line and the object and measure that distance to the red dot, which is the origin. And what this does is, so what I described is steps 1, 2, 3 in the top row. And what it does is it produces step 4, right? We've now turned a two-dimensional shape into a complex sinusoid wave, in which is just a pair of coordinates, right? You have the x-axis is the grid line position, and the y-axis is distance, right? And so in this case, if the crescent object you see at the top, the grid lines, each grid line intersects the object twice, right? So what we do is for all intersections on the same grid line, we sum them into one value, which is why if you see in step 4, right, there's only one dot for every x position, right? And so the innovation here is turning a two-dimensional shape into a complex sinusoid wave, right? That's steps 2, 3, and 4. That's the innovation of this approach. And the reason I did this is because there's a very powerful algorithm called the fast Fourier transform, particularly in engineering, that decomposes complex sound waves into its individual simpler wave components, right? And that's, invented by Joseph Fourier. It's been very powerful and useful in many aspects of engineering and image analysis. And I wanted to turn two-dimensional shapes into a sinusoid wave, into a pattern that the fast Fourier transform can see, but derive a pattern that still captures spatial context in a way that's better than before. Right? Because previously, before you would upload the entire image into the fast Fourier transform, what the computer does is it turns the image, individual rows of pixels, it just lines them up into one very long, right, row of pixels, right? Which works. The Fourier transform sees that, but you lose so much context of the shape in the image. So I invented the LCPC transform, which is steps 2, 3, and 4, so that we can turn shapes into sinusoid waves, but yet capture more spatial context than before. And so at step 4, if you apply the fast Fourier transform, what you get is step 5. That is a frequency plot. And this frequency plot is a multidimensional representation of the image you started with. That is the step 1 image of the crescent moon. And though step 5, it's multidimensional, meaning there's here 25 bins representing the shape you began with. And though it seems complicated, which it is, but herein is the secrets to subtle shape differences, because we can now create an index from this plot in graph step 5, right, to produce a scalar value, right? That is one number to represent the shape that we began with, right? And so, and the simplest way of doing that is to sum all bins, 25 bins in step 5 together into one number. Or you can just divide, say, bin 1 over bin 2, right? So there are many indices in medicine, throughout all of medicine. In particular, the most popular one is body mass index, BMI, right? So an index is an educated guess. It doesn't have to work in every situation. As we know, BMI for very athletic, professional athletes, muscular athletes, their BMI suggests that they are obese, right? So in extreme cases, indices don't always make sense, but there are ranges in which they do make sense and they're quite useful, right, as screening tools, whatnot. And so there are ways to turn the complex data in step 5 into a scalar value, which is one number, right? And scalar values are good for medicine because that's how humans like to understand the world, scalar values, right? But it's also good for machine learning because it makes it easier to build a machine learning model if each feature is represented by just one number, okay? So this is the approach that we apply to all the data that I will be presenting in the coming slides. I'd like to point out that this approach of measuring an index is quite useful for measuring very chaotic systems, right? And here I presented the example of this micrograph of these fibers, right, zoomed in. And I like to point out that there's a difference between chaotic and random, because chaotic systems can have recurring microstructures that we can repeatedly and objectively measure, while randomness doesn't. And this assumption is the difference between finding very interesting things in your data, right, versus relying on somebody else to find it, right, because you just didn't know where to look. And so for a system like this, what you could do is you can outline objectively, right? In this case, in the middle panel, I outlined just either the left edge of the fiber or the bottom edge in red. And in the panel on the right, I extracted those shapes, right? And in this case, if we apply the parallel grid system, what we can derive is an index of either texture or density, depending on how you interpret it, right? And so we can actually quantify very chaotic systems and show that actually they are objectively and quantitatively stratifiable in this case, right? You'll get a very different number from the bottom panel versus the top panel in terms of an index, right? So the idea of an index is very useful in pathology and in epidemiology and many throughout many practices of medicine, actually. And it's quite useful in measuring complex systems. And Shape Genie is another way to measure complexity in 2 dimensions. And this is similar to this same principle I like to point out, because this assumption it's often misunderstood, because we're not used to measuring very complicated things. When we look at it, if the brain can't make sense of it, right, we think that it's haphazard and to the point of being random. And so it's hard to really measure a random system, right? What's the point of measuring something that's random? And it all comes down to assumption. But this assumption is actually quite fundamental in science. I mean, if you think about in high school courses where we teach how to measure the murkiness of a vial of water, right? How muddy it is. Or even in the biochemistry lab, when we do Western blotting, we measure the concentration of protein in your liquid, right? We do what's called the Bradford assay, right? And so when you have a vial of liquid, it's, you cannot predict the motion of every single, particle in it that's muddying up the water, right? But what you can do is shine a light through that vial and measure how much light either passes through or does not pass through. And so at the population level, right, you can actually measure a property of that whole system by shining light through it, right? So that is the same principle that we apply here in image analysis, in measuring an index of the complexity and chaos in our system, right? Though we cannot make sense of individual particles, right, we can measure the system as a whole, and it works amazingly, actually. And another very special thing about Shape Genie that allows us to do, and this is very unique to Shape Genie, is it allows us to finally be able to measure shape apart from size. Previously, this has not been possible because the moment you resize an object, you've changed its volume, you've changed its area, you've changed its perimeter. For example, on the panel on the left are these 2 objects that are the same shape, right, but actually different sizes, as you can see the difference. What Shape Genie allows us to do is to normalize, right, both shapes to be, in this case, the same height based on the longest internal length. And in this way, when we resize both objects to be the same height, while constraining the height to width, the aspect ratio, so we don't skew the object, right, we can now measure their shape apart from their size, right? And this has, every time we apply this to a biological system, we find amazing things. And it also works in physical sciences, material science. But this is something very special to Shape Genie, and is something that I will show you a bit later of how we applied it. But it's allowed us to measure a new subdimension of spatial information that we just couldn't before. But we know makes sense because just because size changes doesn't mean shape changes, and vice versa. So I just want to point out that the software Shape Genie, it's available, and we created free long-term accounts. These are legitimate long-term accounts, so that students and scientists can learn it and use it. We do limit your monthly capacity, how many images you can process per month. in order not to go bankrupt, because we are a for-profit company that needs to be sustainable. But we also created a tutorial videos for you to learn it, as you can see in the link below. So now I begin to talk about how we applied Shape Genie to study Alzheimer's and dementia, and how we were able to capture spatial context in ways that were not possible until now. And so in Alzheimer's, when we study the structure of Alzheimer's, the structure of the brain, excuse me, in structural MRI, right, one of the compartments affected by Alzheimer's are the lateral ventricles, right, these caverns in the brain that contain cerebral spinal fluid. And as you age normally, these caverns enlarge and change shape. But if you have Alzheimer's, they enlarge and change shape even more dramatically. And so here on the right, we measured the shape of this cavern, and it's two compartments because this is a three-dimensional shape. It's like a U shape. And so when you take a 2D slice, it cuts that cavern into two compartments, but it's the same compartment, lateral ventricle. And so when we measured the shape using the parallel grid system, we found a big difference between cognitively normal, that's CN, right, patients versus Alzheimer's patients, which is represented by AD. However, when we measured the same shape in MCI, which is mild cognitive impairment, right, that is some consider the pre-version of Alzheimer's, though not always the case, we found a very weak difference. And to me, I was very unsatisfied, because visually, right, like a pathologist or radiologist would, you examine these recurring patterns in these images, and that's how you train your intuition to pick out what is normal, right, what is diseased. And I felt that the difference between MCI and Alzheimer's should be much more different than what the data suggested. And so I spent more time examining these MRI images, and I realized that in Alzheimer's, not only are the ventricles enlarging, they're actually moving away from the midline, as seen, of the brain. And so what I did was then I added an anatomical marker, this green line right here on the midline. So in every patient, I added this green line of the same length, right? But it's always in the same objective position, that it's on the midline, and it shares the same base as the bottom of the object I'm measuring. And so what this does is it shifts the reference line, the dotted line, okay, to the left of the green line. So that means the outline of the caverns are further from the dotted line now, right? And this simple step, objectively applied to every single patient, finally gave us the numbers that made sense of what we saw with our eyes. That we finally found a large difference between MCI and Alzheimer's, right? Just as we see visually, right? And so I like to point this out because, as far as I know, this is the first and only technology that can capture anatomical or spatial context, right? In this case, anatomical context, this way, right? That not only are the volume and the size and shape of the ventricles changing due to Alzheimer's and MCI, right? But it's changing relative to the midline of the brain. And so, yeah, and just point out that when we do this, we have to make sure to be objective, right? We add the same amount of spatial information, this green line of the same length, right, in the same position in every patient, so that we don't, add bias to the data. And here an example. In this case, we measured the shape of the same ventricles, but in a different slice of the brain. And as you can see here in blue and orange, blue is cognitively normal, orange is Alzheimer's. And as you can see, for every half decade of life, right, there's a difference between the shape magnitude of these caverns in Alzheimer's versus cognitively normal patients. And here I want to highlight the age bins, right? They span only 5 years of life, half decades. As you can see, that the brain is dynamically changing, right? Even as we age normally, the blue boxes, right? But when you add on Alzheimer's, they change even more dramatically. And I'm going to highlight this because all of our data shows this pattern of a half decade dynamic change. And so all of our machine learning models that classify Alzheimer's, the age bin of those models never span more than 5 years, right? Because in our data, if you look as you see in front of you, it makes no sense to combine age bins beyond 10 years or beyond 5 years, because then you would just drown out the difference that you see, in each half decade. And I want to point this out because if you look at the published papers out there that, do machine learning and AI on, structural variation of brain MRIs for Alzheimer's, their age bins span 10 to 20 plus years. And they do that probably because they need a large sample size in order to get a significant difference so that their machine learning model is 90% accurate, right? Yet for us, we would never have an age bin span more than 5 years. So what that tells you is that what we measure through Shape Genie is much more precise than what the standard method can see. And by standard method, I mean volume, surface area, and area, right? And perimeter, right? All our standard methods of measuring spatial information. I want to point out, since this is an audience of clinicians, that this 5-year window of dynamic changes has been independently reported by other people, but also by us. And then when we studied bipolar disorder, we identified, these gyri that you see in panel A outlined in red, right? The shape of the inner edge of the gray matter. That when we measured this shape, right, again with that orange line this time representing distance from the midline of the brain, when we measured this shape, we're able to stratify quantitatively and objectively the difference between normal patients, cognitively normal patients, versus those suffering from bipolar disorder. And as you can see, transitioning from the orange box to the gray box, right? In males, there's a sinusoid pattern. If you look at the averages, right, that there is a periodicity of about every 5 years of this feature, in the brain. But in patients suffering from bipolar disorder, right, when you go into the age of beyond 30, or transition from 25 to 30, there is a major change, right? The sinusoid pattern becomes a sigmoidal or parasigmoidal pattern. And this makes sense of the median age of diagnosis of 25, right? Something special about the age 25 in males in which, bipolar disorder seems to flare up, right? And we found structural evidence of that, right, through these brain folds. And I just like to point out that this 5-year periodicity has been reported independently of us long before we started the project. And one example is this paper in which they measured this notch in the corpus callosum of the brain and the prevalence of people who have a notch in the front, in the back, or 2 notches. And here in this paper, they fit a straight line to the data. But if you zoom in, as I've done on the right, and you connect the dots, you clearly see that a sinusoid pattern, a periodicity, is a much better fit, actually. And this explains why there are squared values on the left are quite suboptimal, in my opinion, because a straight line doesn't fit the data well, right? And if on the right side, if you look at the dotted box, that's the window in which that captures the age of our bipolar disorder patients, 20 to 40. And clearly there is a 5-year periodicity, right? And I understand that this audience is an audience of clinicians too. And so, and you are naturally skeptical of new findings that you haven't seen before. So I just want to point out that we're not the first ones to report that in the brain. There are dynamic changes, right? Even in healthy situations of just normal brain structure that has a periodic nature, that is. All right, switching gears back to the brain. And back to Alzheimer's. And so we work with archive data of MRIs, standard gold standard data sets in the field. And if you've ever worked with archive MRI data, you realize that when you load the data, the orientation of the patient in terms of where they're looking at is different, right? A patient can look, be looking up, down, to the left, to the right. And the thing is, if you're measuring volume, it doesn't matter where the patient is looking, whether you look to the left or to right. The volume of your brain doesn't change, right? But the way Shape Genie measures shape, the orientation matters a lot. And so we realized this when we started measuring shapes in the brains, as I'll show you. And we realized that how sensitive orientation is, or how sensitive the results are based on how you orient the shape to begin with. And though this extra step, it can seem cumbersome and seem like a weakness of Shape Genie, I hope to show you in the next 10 minutes that this fussiness is actually the greatest strength of Shape Genie. And so in order to objectively measure shapes, we had to derive a coordinate system where anybody can follow and orient their data in the same way to extract the same data. And we call this the BSI coordinate system. And it's quite simple in that you align the brain in 3 views, right? First, step 1, in the axial view, you align the brain vertically. Okay, and then in step 2, in the coronal view, you also align it vertically such that the midline of the brain is vertical. And then in step 3, you have to find an objective way to align the sagittal view. And we did this by defining a line that has to intersect 2 points. In panel C, you see the blue line. That is the base of the orbital frontal gyrus. Actually, in this slide, is more like the straight gyrus. Okay, and then the red line, right, is the apex of what we call the inferior colliculus. And so these 2 lines, right, need to be on the same, or these 2 points need to be on the same horizontal line. And that's how we objectively orient, the brain in this system, which we call the OFIC or OFIC triplets. Now, this is one of many possible combinations of rotations, but this is the first one that we came up with and is the one that we use for the data that I'll be showing you. So in this way, using this system, anybody from any lab around the world studying the same data or even different data can align patients in the same way such that the data they will derive is consistent, right, based on Shape Genie. And so let me start giving examples of the shapes we measured right after we orient the brains in this way. So this particular feature is the shape of the roof of the lateral ventricle. Okay, again, these caverns that contain cerebrospinal fluid. Okay, but in this sagittal view, as you see in the image. And then we had to derive an objective way because we worked in 2 dimensions. That is, we only measured 1 slice per patient, right? And we did that just for efficacy. We could do it in 3D, but I did it in 2D for simplicity, for speed, computational speed, and just to show you how effective our approach is, that we can do it in 2D and match what people find in 3D. And so here, moving from, after you orient the brain, moving from lateral, right, to medial, okay, in this sagittal view, right, eventually you will see the shape of the lateral ventricle open up, right? That is that black cavern right around the ear, behind the eye. And eventually you also run into this little gray bump, that is the hippocampus, right? And so the way to objectively pick the same slice in every patient, right, is to move from, in this view, from lateral to medial, and to stop, right, at the first slice that shows the hippocampus. So through this way, we can pick the same objective slice in every single patient. And when we extract, we outline the shape, the roof of this lateral ventricle, as is seen in panel B, and then in panel C, we apply the parallel grid system, right? And with the reference line, the dotted line, always being the line that connects the mouth of that roof. And when we do this, this is the results that we get. Again, cognitively normal is blue, Alzheimer's is orange. As you can see, every half decade of life, we get very robust separation quantitatively. And here is a second feature that we measured for Alzheimer's, right? This is the axial view of the lateral ventricles, but near the back of the brain. And how did we find objectively the same slice in every patient? Well, moving from anterior to posterior, the slice of interest, right, is the slice in which the apex of the cerebellum, here in blue, right, reaches its tallest point. Okay, the first slice in which it reaches its tallest point, that's the slice of interest. So using that anatomical marker, we're able to objectively pick out the same slice in each patient. And then we measure the shapes of these caverns, the left and right lateral ventricles. And again, in this case, we realize that when we add in some extra spatial information, that is a line capped on the midline, in this case is yellow, right? This extra piece of spatial information objectively applied to every single patient, what allowed us to clearly separate MCI versus Alzheimer's, right? MCI, again, is mild cognitive impairment, right, versus Alzheimer's. And these numbers then match what we see visually, right, in the MRI. Even though if you were to measure volume, the difference would be very minor. And so here's the data. Again, you have a 5-year periodicity and robust differences between normal and Alzheimer's patients. Right. Next is, okay, we measured the shape of this cavern in red here. And this is the shape, the space left after the hippocampus shrinks. So Alzheimer's affects the hippocampus, right? The place where memories originate. And as we age normally, the hippocampus shrinks. But with Alzheimer's, it shrinks even faster. And when it shrinks, it leaves an open space. And for us, what we measure is this open space instead of measuring the hippocampus. And in order to pick, in this case, the same slice with every patient, right, we looked at the axial or the coronal view in this case, and moving from anterior to posterior. The slice of interest is the first slice in which you see the surface of the pons, right, which is these two knobs in the blue square. And that's how we picked out the same objective slice in every patient. And when we measure this empty space that is left behind by a shrinking hippocampus, again, we see a difference between cognitively normal patients and Alzheimer's patients, right? Every half decade of life in the ranges that we measured. So applying, okay. And so before I go on to the results of our machine learning model based on these features, I want to talk about a problem that we encountered that the other people in the field have likely encountered, or definitely have encountered, and how we overcame that. And hopefully this might be useful to your work. And so as we were measuring the same features in different data sets out there, different archive data sets, right here, the graph shows the data set from called ADNI and another data set from the NACC. So these are two gold standard data sets archived for neuroscientists to study. And when we measured these features, we found a difference between the blue and orange, right? Normal versus Alzheimer's. But the problem was that the scatter of the orange box in NACC data, the scatter of the Alzheimer's patients, was very similar to the scatter of the cognitively normal patients in another data set. And this was highly problematic because this means that if we build a machine learning model on the ADNI data, it would completely fail to identify Alzheimer's in the NACC data, right? And this made me realize why it's been so hard, why no one has been able to develop and commercialize a classification model on structural MRI for Alzheimer's, because there's a lot of natural biological variation, just that really creates a lot of noise, right? And of course, this is highly troubling to us because we want to build a machine learning model that works across all data sets and all populations, or all demographics around the world. And so in thinking about how to overcome this, I was reminded of my work in gene expression analysis, that is microarray analysis. And this was, of course, before RNA sequencing replaced microarray analysis as sort of dominant method. But in gene expression analysis, we run into the same problem, as in there are variations in data sets from different populations of the world. And this makes sense, actually, this difference in the effect. In this case, it's a morphological feature of populations, because the most popular risk gene for Alzheimer's is the ApoE4 allele. And we have found that the prevalence of the ApoE4 allele actually, it varies according to latitude, right? Global latitude, geographic latitude, right? Due to gene pools, whatnot. So actually, it's not surprising that one population would have a different difference between normal and Alzheimer's compared to another population. So the way we overcame this is the same method that gene expression analysts use. That is, you have to find a housekeeping factor to normalize different data sets, right? And so in this case, we looked at our data and we realized that a one possible normalization factor would be the feature called BSI 50, shown on the right. And that is the shape of the cavern left over after the hippocampus shrinks. And so what we did is for every patient, whatever feature we measured, we divided that number by the area of this cavern outlined in red in the image on the right. Right, so it's a housekeeping factor. People do that in gene expression analysis. They do that in Western blotting too, right? It's housekeeping gene. Typically, it's beta-actin or GAPDH. Now, I'm not going to get into the definition of a housekeeping gene because if you study the cytoskeleton or metabolism, you would argue that beta-actin and GAPDH are not housekeeping genes. They're actually very important genes, but that's a different topic for a different debate. But suffice it to say that this is a standard method of normalizing data. We applied it to our case of morphological differences, and it also worked. So to show you the results, when we normalize the data, panel B shows that, now we're able to develop a model that works not only just on the ADNI data set, but on the NACC data set. Because now if you look at the scatter of the Alzheimer's in NACC data, it's different than the scatter of the normal patients in the ADNI data, right? And so normalizing the data allows us to then develop a model that works across multiple data sets that were captured by, at different times by different laboratories from different parts of the world, right? Representing different demographics of people. And so this method has allowed us to now develop a model that works across, 3 different data sets, which is very important because in machine learning, one of the major problems is overfitting, particularly in medical AI, right? That is, one hospital develops a model that's highly accurate on their data, but when they test their model out on a neighboring, hospital's data, which could be as close as 5 miles away, that model suddenly drops from 90% accuracy to 65% accuracy, which is highly problematic, right? And there's many reasons why. It could be due to the scanner, the type of scanner you have, the resolution of your MRI machine, how your data is stored, right? How the metadata is stored, whatnot. But the more biological reasons is because there's just natural variation, right? In the population that you serve. So this method allows us to normalize the data and develop a model that works across different demographics. And I want to present this because it might be useful to someone in the audience who is doing something similar, right, with machine learning and finding geographic and perhaps genetic differences in their data. So at this point, our machine learning model for Alzheimer's is 95% accurate, and it's has a sensitivity of 75%. And sensitivity is how often we're correct in how often we are correctly predicting Alzheimer's really is Alzheimer's, right? And so this number is significant because this is what the top papers in the field, this is their level of sensitivity, right? 75%. And of course, our goal is to eventually we will break 75% and hopefully reach near 90 in our sensitivity. But I point this out because already using our approach, we've attained the accuracy and sensitivity of the best papers, right, published in the field at this point. But here's the amazing thing about our technology. One, I like to wow data scientists in the audience by telling them that to derive this level of accuracy and sensitivity, we use only 3 slices per MRI stack, right? Because we don't do volume, right? We pick 3 independent 2-dimensional slices, and they're independent, meaning they're far from each other. So if you were to do a 3D rendering, what you get is a block of cheese, right? So you can't even do a coherent 3D rendering with these 3 independent slices, right? But with just 3 slices, and I limited to 3 just to show the efficacy of how effective Shape Genie is, right? But we'll definitely be adding more slices to the left and right of the slices that we do pick, right? To increase the accuracy of our model, right? But we obtained 95% accuracy using just 3 independent slices per MRI stack, meaning there's much more room to improve our a model. We far from maxed out the information that's there in the data. And secondly, this often drops the jaws of every data scientist I talk to, right? So we train our model on only 20% of our data, and we test that model on the remaining 80%. And this is the opposite of what's called the 80-20 split. We do a 20-80 split, right? So the rule of thumb in data science is you have, say 100 patients, right? And the rule of thumb is to train your model on 80% of those patients, and then you test it on the remaining 20%. But this rule of thumb is based on two assumptions. One, it assumes that your data is inherently noisy, which data often is. And two, it assumes that the readout between your two groups, say normal versus affected, is small. So based on those two assumptions, it makes a lot of sense to train on the larger amount than you test on. But we purposely switched the ratio because our data is so clean, right? We get very clean data, as I've shown you on the graphs on the previous slides, such that when we train on only 20, right, we still get 95% accuracy when we test on 80%, right? And this is quite significant because it drops the jaws of every single data science I talk to. And we do it on purpose, right? Just to show you the efficacy of what ShapeGenie can do. And so we're in process of submitting a grant to the National Science Foundation to develop this model for males and females. And we will also attempt to do so for Hispanic populations as opposed to non-Hispanic white patients.
Time is running out.
Okay. All right. And then and how many minutes do I have left?
Yeah, three slides left.
Okay. So on the topic of dementia, so using ShapeGenie, we reinvented what's called the clock draw test for detecting dementia. Mm-hmm. And this app will be available on the App Store in a matter of less than 2 weeks for you to download. And we created a free version, which allows you to analyze your clock once a month. And the clock draw test is a classical test to detect dementia based on how a person draws a clock, as you can see here in the image. But it also detects traumatic brain injury and psychosis. And so we're able to do this because we measure pure shape. So it doesn't matter how the person draws a clock each time, because nobody draws an identical clock each time, but we can resize it to measure its pure shape. So therefore, we can now measure objectively the shape of clocks drawn over time. And here is a hypothetical series of clocks going from normal to highly abnormal. And so what the patient can do is upload the drawing of the clock and get an objective score report. And it's objective now because we have established ranges of what is normal and abnormal in terms of a clock. Mm-hmm. And this allows us to do monthly analytics. So with monthly analytics, the patient can see, okay, are my scores reaching the red zone, which is the abnormal zone? And because this is analytics, that is graphical data, we can create automated email and text alerts to notify the patient and their doctor and their family members that there should be a follow-up because something is going on. So this will allow us to detect dementia early, which allows people to change their behavior, diet, and exercise to slow down dementia. But it also prevents a lot of unnecessary emergency room visits due to accidents, such as cooking accidents and falling and breaking bones. Mm-hmm. And so this is an example of ShapeGenie applied to hand drawing data, right, to solve reduce the burden of dementia. And so, yeah, so now since my time is up, I think I'll end the talk there, and I'd be happy to take any questions from the audience.
Thank you very much, David. This is really very impressive. To be honest, I'm a little bit smashed. I know your data. You have been so kind sending me that earlier. But I stay honest also as a scientist. We are used primarily to think and to view in an anatomical way. By simplification, getting away from volume, it's a kind of simplification, correct me if I'm wrong. Now we get in a complete different direction. And I directly had many thoughts going into my mind. So if we, for example, measuring these 5 years changes scales, and if you compare, just to make an example, if you compare 2020 measurements to 2025 measurements. In terms of the data, is the standard deviation which you showed in your box, is that decreasing? Because this would give us the information. It's a kind of learning system, and the data improve as older they get. Correct? Or am I wrong?
Yeah, what you're asking is actually two questions in one. And so, yes, a model, I mean, the data we have is cross-sectional, meaning it's not longitudinal, right? It's cross-sectional. Each patient is different. So we don't know for each patient how quickly they get to that, how quickly their shape changes before they get to that point. But nonetheless, most machine learning model data out there are cross-sectional, right? And of course, as long as you validate them, then you know they work. But in terms of yeah, I mean, so the brain changes that naturally happen, right, at different points, that has to be integrated into the model such that the model, is able to capture, the risk at different age points. So our models thus far, that's why we have a model for each half decade of life, right? So we wouldn't, if you're given your age and gender, right, we have gender specific or sex specific and age specific models. So we would have to apply a model that's specific to your category, right, in order to either trust the classification, right, or trust any risk prediction that comes from that classification.
Okay, but if you have cross-sectional data, what would you bring it to another point, but to keep on that theme, because I think this will be of high value later on to value your work. When scientists value your work, they will ask you in 2-3 years, did you have a comparison of the data 2-3 years ago from various leading scientists who placed the midline line, the green one? So is it possible for you to get an information or an impression? Is there an inter-observer variability when you define that? Or is that completely excluded?
Oh yeah, so just like any method, we would have to, have it done by different laboratories, right?
Right.
Read it, but in machine learning, the inherent way to do that is keep validating your model, right? Okay. And so that, and you would know. So it's the same approach. A different approach is the same goal, right?
But we will see it in the future, correct? We don't see it now, but but this will be very important. And I'm pretty sure you will have a high value publication in terms of that. Because your approach, correct me if I'm wrong, always, okay? Just interrupt me if I'm wrong, okay? This is my principle. And there are no stupid questions, there are only stupid answers from my point of view. Point to me is that I get the impression that your approach tries to improve the value of an imaging method. Now you go here and showed us radio morphological imaging, but this could also mean that one day you could also use that in terms of single cell analysis.
Absolutely.
This would mean you could use that for hematology. Why today we use phenotypization for hematology to get the information, is which kind of lymphoma, leukemia, blah, blah, blah. So this would be another approach. Another thing which which I was intrigued when you lectured was you used to compare the information about APOE4, correct? Right. Did you ever use it with tau?
No, I mean, I mentioned APOE4 as an example of how that, popular gene, the prevalence of it varies based on geographical location. I mean, whether or not tau deposit likely varies too, because we know, I'm a big fan of research out of UCSD, Sarah Banks and Aaron Sunderman. They found that in women, the deposition of tau is different compared to men in Alzheimer's. And in fact, women, the medial temporal lobe can handle more tau damage, before verbal reasoning is affected. And they say, therefore, if you're diagnosing Alzheimer's in women, you should not be using verbal reasoning tests because you're going to underdiagnose them, right? So I can't cite the data, but given how tau works, I'm pretty sure that it varies by geography too.
Have it in mind, we have a very important scientist in terms of Alzheimer research and science, Professor Franz Teuring from the Berlin Society Scientific Society. He will give his lecture about Alzheimer and the amyloid dogma, because he's a tau researcher. And we all know that the amyloid dogma is nonsense, because a symptom was measured and was transformed to be causative. So, and now comes the point. We are in the same situation in terms of epigenetics and genetics. A symptom was usually measured, but it was transformed to be causative. And that is a huge, huge mistake during the last 70 years. And the mass of scientists do not want to see that, because they are in with blinker, without thinking out of the box, going in their way.
Yeah.
You measured the overall accuracy, but I have to insist, because I was trained during my PhD work by a very, very intelligent French mathematician. She won one of the highest prizes in mathematics in France. And she trained me in terms of performing ROC analysis, receiving curve analysis, which is necessary when you want to measure accuracy, sensitivity. But you did not provide us the information about specificity, positive predictive value, and negative predictive value. So I'm sorry, I have to insist and to ask you that.
Yes, no problem. Yeah, and we have that, and our specificity is 90%, right? And then our sensitivity.
So you have a sensitivity of 75% with a specificity of 95%, correct?
Yeah, accuracy.
Because
95%.
what most people do not understand, to make the decision where the threshold is in receiving curve analysis is a clinical decision. It's not a decision by a program. You have to sit there, you have to see the data, and you have to look at the values, and you have to make the decision, what do I want to have? And then you make the decision which threshold you want to have, and then you get the information with sensitivity, specificity, PPV, and NPV. That is the reason why I'm asking, because it's still not a machine decision. It's a clinical decision by the scientists. And this is what I would say 90% do not understand.
Yeah. And so, I mean, it's fault of mine that I didn't show that. Typically, it's in the field of data science, what people care about in terms of the gold standard to break is the specificity of breaking 75%. But yeah, you're absolutely right.
Okay. So what I found extremely also interesting, to remind me about my Helmholtz research, performing RT-PCR, when you brought it up and said, no, we don't take a housekeeping gene where we use that. Now we take a housekeeping factor like in RT-PCR. So again, to understand it, you define what the housekeeping factor is. What are the decision-making variables? What could be a housekeeping factor to get to the direction that you all afterwards have a normalization of the data? How do you make the decision?
That's a great question. Yeah. So typically, you pick a gene or a factor that doesn't change, right? Typically in your treatment group versus your control, right? That's relatively stable, doesn't change as much, right? And that is typically the approach to the housekeeping, right? And so typically, that's why in gene expression, there's a handful of common housekeeping genes that everybody uses that people pick from. But
although I have to interrupt, although that we know that if you look at the half-life time of proteins, it's so less and so small that usually you have to re-ask the values of the results, to be honest.
Exactly. So which is the debate of, are housekeeping genes really housekeeping genes? Because for for labs that study the housekeeping genes as the main gene of their career interest, to them they're offended that you called it a housekeeping gene, right? As in a gene that doesn't not important, doesn't change, right?
Yeah.
But so it is a methodological technique that does work, and of course we trust it because we keep validating our model, right? That's how we know, right? We test them on new data, right? And the predictions maintain the accuracy. Then we just trust the methodology without getting into the weeds of what really is why you call that gene. Yeah.
But I have to insist. So you transform that model from molecular biology to your approach. And how do you go to the direction to decide what is your personal, in that kind of research, what you do to make the decision? Do you have a list and say, I want to have this, 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 this? How do you come to the decision making? Is there an algorithm which you created that you declare this will be the housekeeping factor?
Yeah, it's an intuitive algorithm that is just our reasoning, right? And that is, we looked at the data of that particular housekeeping feature across the same data sets, right?
Okay.
And we saw that it didn't vary much, across, and so we felt that would be a good choice. To be housekeeping factor, because all the other ones varied a lot, right? Between controlled versus whatnot affected, and so that was the rationale.
This would be highly interesting because we have a wonderful colleague. In the future, you're going to have a talk. This is Professor Masaharu Seno from Japan, and he's doing stem cell work. And to bring people like you together with him, or with Franz Peuring, who does tau work in Alzheimer, I think this could be huge in the future. But I think it's a long way to go. So I think you're a pioneer to do that, and I'm very, very thankful, very proud, very privileged and honored that you found the time to get up early. And that especially in San Francisco Bay Area, which is very, very uncommon for scientists. Thank you very much. I really highly appreciate it. So there are no questions right now anymore. I think people have to think about that. We close the session and we want to thank you. We give you our deep gratitude and our deep thanks to the USA. God bless you. Have a good day.
Thank you, Bjorn. Thank you, everybody.
Bye-bye.